So just as we're waiting for everybody to join, I see we have 124 people online. So thanks everybody for being prompt on this Tuesday morning. My name's John Cumbers, and this is live from the Bay Area, California. And this is a SynBioBeta Town Hall on the COVID-19 pandemic response. We're going to be speaking shortly with Jason Kelly, the CEO of Ginkgo, on the state of the industry, and also on Ginkgo's $25 million towards access to their platform for people who are working on COVID-19 activities. We've got a whole number of people who are joining. I can see Casey from Conagen, Casey Littmeyer, Pia from Blue Yard in Zurich, Chad Pastor, Michigan State, people from Georgia Tech, Cameo Biosciences in the UK, Ellen Jorgensen from New York. Our hearts go out to the people in New York right now. Stradios, Mark Fisher-Colbury, welcome, good morning. Everybody dialing in from Twist in Carlsbad. Hi, Katie Kramer, who was instrumental in setting up our last town hall. We're still seeing a few people dialing in, so thank you all for joining. I'm seeing a lot of CEOs. The CEO from Procarium is Ted Fallman. Welcome, Ted. And Isaac Larkin from University of Chicago. Derek Greenfield from iMicrobes. Excellent. Great to see everybody joining. So continue to type into the chat box. We want to be able to get a sense of how long this each person should be able to speak for. So once we've done further comments with Jason Kelly and Patrick Boyle at Ginkgo, we're going to be talking about what you're doing in response to the pandemic, and particularly if you've got any resources that you want to offer or you need help from anybody. So if you would like to speak, please raise your hand in the app. That's going to help me see how many people we've got to go through, and I'll judge how quickly I cut people off who are going too long when we go through the round robin. Alfonso Jaramillo, welcome from Warwick in the UK, and Tasha from SynBioTechnologies in New Jersey, and Johns Hopkins Center for Health Security, Rachel West, welcome. We're also going to be doing another town hall tomorrow with Stefan Bancel, who is the CEO of Moderna. They're going to be talking about the RNA vaccine that went into clinical trial just last Monday in Seattle. And we're also going to be talking on Friday on environmental sampling for the SARS-CoV-2 virus with Chris Mason from Cornell, Emily LaProusse, the CEO of Twist, and Josh Buffetto, the CEO of Chai Bio. I didn't realize this, but in the food prep industry, they don't wait until the food is contaminated with a pathogen before they test it. They're continuously testing the environment in industrial and commercial kitchens to make sure that there's no virus getting anywhere near the food preparation area. And so this Friday, we're going to be talking about what we can do for environmental testing of SARS-CoV-2. So with that, I would just want to remind you that this is going to be a live Q&A. We have 150 people joining, and we're going to try to get through as many questions as we can. And in particular, if you want to speak, then please raise your hand. So far we have 4 people who've raised their hand who want to talk. And after we get through the discussion with Jason about Ginkgo's response, then we're going to be able to go do a round robin. I will pass the virtual mic to you, and we will go around and ask what you're doing in terms of the response, what you need help with, and how other people can help you. So that's great. I see we have 6 people who are going to be doing that so far. So with that, over to Jason Kelly. Jason, welcome to SynBioBeta Live. Thank you for joining us. I know that you must be extremely busy. It looks like you're working from home today. If you can just give us an update of how things are looking in Boston and what's the status right now of Ginkgo.
Yeah. So, I mean, Boston, like many other cities, is moving to shut down orders. Closed restaurant and bars last week. Now it's all non-essential businesses. Ginkgo's still operating under that order in a capacity around COVID-19 in particular. What I would say, I guess, is it's not too different here. I think that it is around the entire world, where I think you have a lot of people very stressed out and concerned about this situation. And I think, Scott Gottlieb had a pretty nice series of tweets maybe a couple of days ago, basically explaining, look, we're basically taking an economic hit right now to try to avoid this. And that's creating other pain. And if we're going to ever get out of this, we need a technological solution in the long run. And so I think that to me is
former head of the FDA.
That's right. Thank you. Yeah, here in the U.S. And, I think that to me gives the synthetic biology community suddenly some agency in all this. I think we have technology that can contribute to vaccines, that can contribute to therapeutics, that can contribute to point of care diagnostics. Those are all things that are, they don't matter. It'd be great to have them this week, but if we don't have them in 3 months, 6 months, a year, this could get real bad. And so I think we are an important part of this, and we should step up as a community in that lane. That's a thing that, no bullshit, we can help with. And so that's what we wanted to do with Ginkgo. We wanted to basically say, look, how can we help it as fast as we could? And that was to essentially open our platform up for free for folks working on COVID-19. And for folks that don't know much about Ginkgo, our whole model is to operate in a platform footing and a partnership footing. In other words, we don't develop therapeutics. We partner with people that develop therapeutics. We don't, we work in things like the ag industry. We don't develop new agricultural products. We partner with companies like Bayer to do that, right? And so in this regard, we're actually well set up to enable the folks on the call today who might be developing a new therapeutic or vaccine, point of care diagnostic that could make use of a lot of cell engineering. And so Patrick will speak to some of the capabilities we have, but we basically said, look, for $25 million worth of platform, we'll go ahead and do that. So we're starting these projects for free. You can email at covid-response@ginkgobioworks.com, and we'll quickly see if there's a platform fit and have our technical folks talk to yours. And if there is, we'll just get started. Won't cost you anything. And so that's sort of what we felt we could do. And I'm sure many of the folks on this call also have things they could do, whether it's developing the product or if they're a company like Ginkgo that has a technology, maybe just getting that in the hands of the researchers doing this work. The other thing I'll say is we are seeing a lot of, there's a lot of private funding sources that want to back COVID-19. I think they don't know how to think about this stuff. They're not technical. So another thing we're trying to do is bridge those types of private funders with promising projects. So where we see the opportunity, we'll try to do that also, just as a second type of thing we're doing in addition to the technical platform. And then the last thing I'll say, and I'll hand it to Patrick to share the details on the technology. Look, we've been in the startup ecosystem, John, you and I, around synthetic biology for pushing a decade now. This is gonna be a really hard time, and I just I think remember we are a community of startups here. If you're feeling like you're getting squeezed on this, I think you should reach out to other members of the synthetic biology community. I'm available. Emails Jason@ginkgobioworks.com. Happy to talk about anything when it comes to startups, fundraising, or otherwise. Shoot me an email. So just wanted to get that out for the community. And that's it for me. And I was going to hand it to Patrick to talk about some of the technical details.
Great. So just before we do that, that's a good point. We actually have a poll right now. We'd like to feel the pulse of the SynBioBeta community. So if we can bring up that poll right now, and we just want to know on a level of 1 to 5, how's your stress level? With low is number 1 and 5 is high. So if you just want to go ahead and fill that out, we're going to
try to
share
check
the
how
results.
loud my children are currently screaming before I fill out this poll. Hold on, let's see.
It's all good, Jason. We have screaming children in the background, so we should be able to begin that poll any minute now. So if you want to get that going, and then we can vote on that. So in the meantime, I just want to remind everybody that this is a live Q&A, and we're going to be taking results from the audience and looking at what you're doing in response to COVID-19, how we can help you and how you can help other people in the community. So if you'd like to raise your hand, then you will be able to see other people who are raising their hands as well. And then we're going to call on people to see how they can contribute to this response. So we're going to be going through the list of attendees, and I see we have 8 people raising their hands. So as soon as we've got through Patrick's comments, then we can do that. So if we can show the results, there we go. So we've got maybe if you can just display those results to everybody. We can see 37% of people on a 4 out of 5 in terms of stress, 33% on a 3 out of 5 for stress. So we got quite a lot of high levels of stress going on right now. So before I pass over to Patrick, then I'd just like to do a de-stress exercise that I was taught recently at a course that I took called Managing Emotions Under Stress. So this may sound a little bit hokey, but cortisol is that neurotransmitter that builds up in your brain when you get stressed. And right now, particularly, I don't know if you've noticed, opening up Twitter can just send your cortisol levels crazy, and it takes over an hour to get rid of that. and the half-life, I can't remember what the half-life of cortisol. So no matter what you're thinking in your brain, there's a chemical that's pulsing through your blood that's telling you that you're stressed. So we're just going to do a very quick de-stress exercise for a minute or so. So if everybody could join me, I just want you to just relax and let your arms go for a second, loosen your shoulders, and please do join me at home, because it'll make the rest of this town hall a lot more pleasurable. And I just want you to breathe in for 3 seconds. And then hold that breath for 3 seconds. And then breathe out for 3 seconds. And we'll just do that 2 more times. So just breathe in for 3 seconds. And just hold it for 3 seconds. And just breathe out for 3 seconds as well. And we're just going to do this one more time. Just breathe in for 3 seconds. And hold for 3 seconds. And then breathe out for 3 seconds. Okay, and if that wasn't introduction enough, over to Patrick Boyle at Ginkgo. Patrick.
Thanks, John. And with the reminder that Twitter increases cortisone levels, I'll let people know that I'm running the Ginkgo Twitter account this week, so I can't take a break from it. But that's another way to reach us if you have questions or comments on our work. Just to reiterate, what Jason was saying, we're really making this a priority at Ginkgo. I think as a community, we're all in this together. Our priority here at Ginkgo is to do what we can to help the community shorten the pandemic and make enduring it easier if we can. So from Ginkgo, I've been leading our technical efforts to develop collaborations and projects in this space. I think one of the most critical things to emphasize, we recognize that many companies and teams out there are working as hard as they can, as quickly as they can, on applications that impact COVID-19. And one of the challenges there in collaborating is that, like we see our COVID-19 projects at Ginkgo, those projects are likely mission critical for all of you as well. So what we're working to do is to help as many teams as we can de-risk their work by getting that work on board our platform. So, knowing that this work is mission critical, how can you work with a collaborator while you're trying to make sure you get this work done? What is the work that Ginkgo can do in parallel with our technology to give you another shot on goal for that project, knowing that timelines are tight and there's a lot of data that the community is trying to gather quickly. Normally we spend a lot more time with partners getting them familiar with our platform and how we can help with their specific projects. I thought I'd run briefly through some of the technologies that we're engaging on with collaborators to give folks of ideas of how we could work together. Obviously, there are a lot of clever folks out there who have very unique approaches to this. So again, think of this as an introduction to the platform, and happy to get deeper with folks as needed. So, myself and a large part of the commercial and technical team here at Ginkgo have been basically on the phone every day over Zoom with various collaborators working on these types of projects. So consider this an intro, and happy to get deeper with everyone as we move forward. I think one of the first things that we announced in this space is opening up our DNA synthesis capability. So under normal circumstances, Ginkgo is the world's largest user of DNA synthesis. So we have a billion base pair supply agreement with Twist, and we also have our own in-house DNA synthesis capability coming from the acquisition of Gen9 about 2 years ago. One of the things that we like about that in-house DNA synthesis capability is that it's complementary to the work at Twist, and we can focus it on custom synthesis of large constructs. So one of the things we're looking at there is, are there labs that need synthesis of constructs up to 10 kb in length? That's a customized service that we offer at Ginkgo. We're also partnered in the synthesis space. for COVID-19. So in collaboration with Drew Endy and the Stanford Bioengineering department, we are synthesizing a lot of the key constructs that we know that researchers need to respond to this. In particular, the first set of things that we're synthesizing there are every open reading frame in the virus and an expression construct with a few different tags that are useful for people who need to do antibody discovery or antigen development research with those constructs. In order to make that available to as much of the community as we can, we're partnered with both the BioWorks Foundation and AdGene. So both of those groups have agreed to deposit and share those sequences with anyone who uses those services. And also for this work, AdGene is lifting its restriction on providing plasmids to industry. So AdGene's repository for COVID-19 work via this collaboration is open to companies as well, which is not how they typically operate. In addition, we've gotten a lot of inbound requests from researchers on custom requests for synthesis. We are fielding those as quickly as we can, and we'll get back to you. If we feel that the standard set of things that we are synthesizing is responsive to the work that you're doing, we'll point you to that resource. But we're also taking on a lot of custom requests as well. Some other capabilities, so moving downstream of synthesis. Ginkgo obviously does a lot of screening. Perhaps unique to Ginkgo in the space is that we're really built around flexible screening. So Ginkgo is often supporting 20 to 30 projects at any one time. That means our screening team is highly skilled at developing assays and deploying them on our automation in a shorter time frame than you might expect. So for example, we've gotten a lot of outreach around groups that are perhaps in silico companies or labs that have some interesting compounds they want to target against enzymes, for example, in the COVID-19 genome. We're working with a lot of those groups to see, can we generally support them by developing the types of compound screens that allow them to actually prove that out in the lab and accelerate development there. We also have some work ongoing in antibody lead optimization. This is work that our mammalian team has been leading since the launch of BioWorks 4. So just to let folks know, if you're not familiar with Ginkgo, we have experience engineering a wide range of microbes, gram-negative bacteria, gram-positive bacteria, yeast, filamentous fungi, and mammalian cells as well. I think for antibody lead optimization, both yeast and mammalian cells are relevant to that work. Our mammalian team is available to consult with groups that may be able to benefit from the work that we can do there. Some unique things that team is doing is building stable strains to handle libraries of antibodies for screening, as well as using our design team to further optimize those antibodies. And then, of course, we have a $150 million agreement with Berkeley Lights to use their Beacon platform, and we're working with them to leverage that platform for antibody improvement via collaborations as well. And then, finally, before I pause, Ginkgo obviously does a lot of full stack strain optimization. So since we have experience in engineering and optimizing various microbial and mammalian strains, we feel that a number of groups that have high priority needs for strain optimization, whether that's protein therapeutics, nucleic acid vaccines, and those types of larger partnerships, those are things that we're actively looking at as well. For example, for nucleic acid vaccine production, E. coli or similar microbes are often the manufacturing host for those products. Ginkgo is really good at leveraging our automated fermentation capacity to quickly optimize processes. And I think that's a great example of the type of work that Ginkgo can take on in parallel with companies that have a lot of other important work to do. So maybe I'll just stop there. It looks like we have some folks who want to ask questions. I'll just say, in addition to dropping messages in the chat, we have an email address set up, covid-response@ginkgobioworks.com. And we have a whole team of folks over on the Ginkgo side who are triaging those emails as quickly as we can and working to get back to everyone as quickly as we can.
Thank you, Patrick. And I also didn't say, but of course you're live from the holodeck there. So can you just describe what we're seeing in the background?
Yeah, I've been having a lot of fun with custom Zoom backgrounds as I've been spending all day on Zoom. This is actually just an image of BioWorks 4. So this is what
you're not actually live there.
looks like. Yeah, no, I'm in my office, but my office is less impressive than than Bioworks 4. So.
Okay, I'd just like to, in this stressful time, bring some joy to people. So maybe you could switch the background to what you had on when we were doing the test just now.
Okay, stand by. I've got a few ones queued up.
Standing by.
Hey, while we're waiting, I'll just say, I think it is important. It is.
We'll go back to Patrick in a second.
Please do. Yeah, go ahead. Go to Patrick.
Okay, just start speaking, Patrick. We'll come back. It'll come back to you.
Oh, just reporting here from the enterprise now. So.
Thank you, Jason. Back to you.
Sorry. Yeah. The only thing I was going to add. Patrick mentioned it, but we're a good source of doing redundant or expansion work on what these teams are already doing. So if they're, I think that can be an important point. If there's a project that's already ongoing, usually have a team really cranking on the lab. You could create a side channel of work or a redundant set of work. I think we're very happy to do that. And also, if you have no other people that should know about this, please do pass this on. The folks on this call have that kind of network that reaches out to these researchers. So please do let people know. Thanks.
Excellent. Thank you, Jason. Thank you, Patrick. Thanks to everybody at Ginkgo for everything that you're doing in enabling your foundry for COVID-19 response. So we're just going to do a mic pass here. I'm going to start with Ted Fahlman. Ted, if you could just introduce yourself and Procarium and say where you're based and talk about this exciting oral vaccine that you're working on.
Thank you, everyone. Greetings from a locked down London, UK. We're an engineering, biology, and immunology company called Procarium. We were the first company to take a bivalent enteric fever vaccine into the clinic. So we're specialists in late preclinical and early clinical development of vaccines. We are a synthetic biology company using Salmonella as a delivery vehicle for oral vaccines. And we're spearheading a consortium of companies, organizations, universities, and key opinion leaders from around the world to develop a orally deliverable vaccine based on our platform, which has already been in 10 clinical trials in the US, UK, Vietnam, and can be scaled to 500-liter culture. And happy to say I spoke with Patrick yesterday and some colleagues speaking to Ginkgo already, and hope we can all put our heads together. Especially happy to hear about your open reading frame project there. I think that's extremely valuable. So good luck on all that. And to all the people listening, humans live long. And
thank you, Ted. Much appreciated, man. Much appreciated, Ted. Ellen, founder of GenSpace in New York City. Tell us what's going on in New York City. How's everything looking and what you're working on?
Oh God, it's crazy. It's a ghost town here. I'm on lockdown. I have been since I got back from Europe a couple of weeks ago. But there are people still coming into our lab because at my startup, Ginkgo Biosciences, we're actually, our mission is to protect supply chains. So we have these custom microbes that contain DNA barcodes that you can mix into food and really apply to any supply chain. And it was sort of a logical jump to doing environmental testing for the SARS-CoV-2 virus. And so we've been rolling that out. We did our first tests last week. And we're offering it to our supply chain customers and also free to local schools and anyone else who might want to, say, check their cleaning procedures, stuff like that. So the other thing I'm involved in, which is very exciting, which I think Isaac might say something about too, is this open COVID-19 project on Joggle, which is just one giant lab, which grew out of the community lab community with Thomas Landrain, who started La Paillasse, the Paris DIY lab. And this is an amazing collaborative platform, and that and the accompanying Slack channels. We've got something like 50,000 people all collaborating on different types of initiatives, everything from validating different test methodologies, diagnostics, to making masks out of and ventilators out of weird things, to community spreading the word type things.
Fantastic.
So, yep. So I'm just gonna put into the chat how to onboard into that effort if you're interested.
Yeah, absolutely. Thank you so much. And Isaac Larkin, if you just want to introduce yourself, and you're calling in from Chicago, I believe.
Yeah, hi everybody. I'm a PhD candidate at Northwestern, and I'm involved in various democratized bioengineering efforts. In particular, relative to this one, the BioBlaze Community Lab and the Free Genes project that's being organized through the BioWorks Foundation. And the should I just mention this? I had a couple questions. Should I ask those now?
Yeah, why don't you just quickly mention what the Free Genes project is and any efforts that they're working on for COVID-19, and then go ahead and ask the question.
Yeah, so the Free Genes project is organized through the BioWorks Foundation, Drew Indy and Keone Gandal, and various other people at Stanford leading it up, but it's also a distributed effort, so I've been contributing to that. And it's basically making synthetic DNA useful genes freely available to anyone who wants to use them to do useful things. And so in particular, a particular relevance to this call, referencing back to what Jason mentioned about the importance of the SynBio community developing, point of care diagnostics. The Free Genes project, we've basically made a spreadsheet of like 100 different enzyme genes that would be basically used in RT-qPCR, in isothermal DNA amplifications, in various enzymes for different types of colorimetric and fluorometric readouts, and we're looking to basically make that collection available in expression vectors to everyone in the world to be able to rapidly prototype, replicate, and scale diagnostics.
Great, excellent. And you had a couple of questions, Isaac?
Yeah. So first question was, I know for Ginkgo or for Jason and Patrick, I know Ginkgo has done a great work with synthesizing all the SARS-CoV-2 coding sequences, and just wanted to know if the Free Genes diagnostic enzyme collection has crossed your radar yet. Are you guys planning to synthesize that as well? And then the second question I had was, I guess for both of them as well. It seems like the rate limiting step in terms of diagnostic throughput is going to end up being like sample processing and nucleic acid extraction. And I think Ginkgo isn't CLIA compliant, but you guys do rapid screening of large numbers of strains, and so I'd imagine you have experience with like high throughput nucleic acid extraction, and could offer insights onto how that could be done both in like high throughput labs and also scaled in more distributed way.
Yeah, so I can take that. Just briefly on the Free Genes project. First of all, yeah, thanks for your support there, and really, wish it was under better circumstances, but it's about time that we got to collaborate on that sort of stuff. We know synthesis started last week on the first batch of things. I can check back specifically on the 100 genes that you specified, but our plan here is to do this in multiple waves. So as we get more feedback from the community, we definitely want to start more synthesis. I mean, in the scheme of the $25 million commitment that we've made, we're not going to break the bank doing this sort of synthesis, and we're optimized for scale. So I'm happy to support the community there. Feel free to be in touch with the folks that you've been talking to at Ginkgo on that, and I can check in on those constructs. But yeah, we'll be doing another round for sure. Mentioning nucleic acid screening and extraction. So we actually just posted something on our website today, and this was we tweeted about this on Twitter as well. We have a pretty unique next generation sequencing capability at Ginkgo. Given the amount of DNA that we're building, we need to routinely sequence thousands of plasmid sequences every day at Ginkgo. So we've been working as an internal project and now with collaborators on can we repurpose that platform to sequence thousands of viral genomes per day? We think we can. We're doing a lot of methods development right now. I'll give a shout out to Twist that sent us their RNA controls to get that work started. That was awesome. And we're also working with CLIA certified labs who can supply us with that material. So, Ginkgo is not planning to do any CLIA certified work, but just providing those viral sequences we think will be a tremendous epidemiological resource. So we're partnering with CLIA approved labs who are doing qPCR. Basically, can we receive the rest of their extracted material on plates, sequence that, and make that available to the community? no promises there. We have a really our entire NGS team and software team and automation team is working every day to get this going. And labs such as Battelle's CLIA approved lab in Ohio is already on board with us to provide samples. That may be happening as soon as this week. And as soon as that method is online, we'll be providing those sequences as best we can, thinking that Ginkgo's capacity on paper should be able to supply 10,000 viral genome sequences per day.
Excellent. Thank you very much. So we've got quite a few people to get through, so we're going to keep moving. If you want to join us, you can raise your hand and you can either come on as a panelist with your video or just join via audio. Just let Kevin know in the background. Attila Chodash, you're in Cambridge, UK as well, I believe. Welcome.
Hello, hi. Can you hear me okay?
We can, yeah.
Great. So thanks, John, for doing all this. So I'm the founder of Cambridge-based H-Curve Limited. And just added to all the external stress that we're experiencing, I just closed our first serious round last week. So my
congratulations.
We're really high. And we try to pivot right now. So our basic expertise is based around deep proteomics aging profiling. So we do protein sequencing from the saliva. That's like 3,000 human proteins abundances and 3,000 oral microbiome bacterial proteins. So for us to search for viruses as well in a targeted proteomics assay context is pretty straightforward. So we put together a library that would measure COVID-19 and a general winter illness panel of rhinoviruses, influenza viruses to have an idea. We targeted unbiased mass spec peptide sequencing. So the accuracy level is going to be very high here. It's not going to be a serological test, but a molecular one on the protein level. And adding to this, based on our aging longevity experience, we can add a robust immunosenescence signal to potentially show who gonna be at higher risk of disease severity based on their already compromised inflammatory immunosenescence signals. Added to this, we could measure bacteria changes, which might, that's the most speculative part, give us a chance to infer back what kind of viruses the organism was exposed to, because that induces changes with co-invaders and all that stuff. And also we have a very, very robust, since you're talking about the saliva, adaptive immunity signals. That's gonna be just generally show you next to the innate immunity or molecular cell senescence and all the other stuff that we're measuring in terms of aging. And my question would be, so right now we're going through the ethical because we need positive samples, saliva samples. So going through the ethical setup to get those. We can already search for the negative samples and assess true negative rate. And my question would be, not a particular platform-based stuff, but for startups that try to pivot right now to do something about this pandemic, would be any stimulus package, which is what I would call basically a donation or someone between business that would help these startups do their work. Thank you.
That's a great idea. Thank you. Anybody want to take that, Jason or Patrick?
Yeah, sure. So I think you're seeing things like, HHS in the United States, this is all 2.5 weeks ago or something. So I assume the view has scoped up since then, but they got approved an $8 billion budget, of which about $3 billion was earmarked for research and development around COVID-19. That those gears are turning, I think. The honest truth, I think, is the government, it's so overwhelming right now to focus on the near-term public health response that is, I think, also which is the right thing to be doing right now. It's just in 6 months, we got to also have all these, we need more point-of-care diagnostics. We need therapeutics, right? And so we need to start those at the same time, but it's a little bit of it's a lot of noise right now. I do think things like that will exist. And there are a lot, of calls going out for proposals, and then that money is going to start to flow, I think, in short order. And then on the private side, I was mentioning that earlier, I think you do see a lot of people, folks in Silicon Valley, elsewhere, that want to give dollars to that, but connecting the dots is hard. And so that's one of the things we'd like to try to do.
Great. And we're going to be having an investor roundtable probably the week after next. And that's a good segue for me to talk about some of the town halls that we have coming up next week. We actually have a number of state of the industry or state of the union town halls that we're going to be doing. Next week, we have Craig Venter is on Tuesday, I believe. Jim Collins is on Wednesday. And George Church is going to be on on Friday. So you'll be getting those dates and emails in your inbox shortly. And then on the Monday following that, we have Drew Endy joining us. So a whole bunch of interesting people are going to be coming on to talk about not just the state of the current pandemic, but also the state of our industry as a whole, and what this means for the future and our interaction between society and biology. So some very deep reaching conversations that we're going to be having. So just as a reminder, this is a live town hall, and if you have something that your company is working on that you want to publicize and tell the community about, just click hand raise in your Zoom, and I'll be dialing you in and making you a panelist. Mike, over to you. Mike Kent, what are you working on and where are you based?
Hi, I'm in Albuquerque and I work for Sandia National Laboratories.
Welcome. Thanks for joining us.
Thanks for putting this on. I do in silico prediction or design of neutralizing antibodies against the spike protein. And we have some capability to test initial predictions for binding, but our budget is limited, and of course there are a lot of possibilities. So I'm looking for.
So I think we lost Mike for a minute there. Andrew, if you want to go ahead, and when Mike's connection improves, he can join back in.
Sure. Thanks so much for putting this together.
You're welcome. Thanks for joining. If you can just introduce yourself and say where you're dialing in from.
Sure. Andrew Satz. I'm from a company where companies evolve, and I'm based in Miami. Similar to Mike, we do computational generation of antibodies. We use evolution as a way to generate those antibodies. And so we actually screen our antibodies at scale to prevent downstream failure. We've pre-screened currently about 2,000 antibodies, and we were sending them to the lab, a bunch of different laboratories to have them produced recombinantly and check binding affinity and initial functional assays that allow us to move quickly into moving them to additional functional testing that'll hopefully allow us to accelerate pretty quickly into animal studies. We're working across a bunch of different targets, including the spike protein, the ACE2, where the virus binds to in the body. I think there's a lot of opportunity to look at other places on the virus itself, looking at the envelope and membrane. So if anybody out there is working on those, one of the things that our technology does, it allows us to generate daughters of existing antibodies. So, and then also pre-screen them and validate them. So if anybody has an existing antibody and they want to amplify their existing antibody or hit some leads, we're happy to work with collaborators on that. We can also computationally screen existing antibodies in a matter of minutes and provide a report. So, anything we do to help the community using our technology, we're happy to support, both in terms of Ginkgo. We'd love to work with you to take some of the additional antibodies that we've generated, have them produced and screened, as well as if you have some that are being generated by you and you want quick computational screening across manufacturability, immunogenicity, thermostability, how well they function, pH. We're happy to utilize our technology. We can do that fairly quickly at scale. So it's something we're happy to do. And because it doesn't take us a lot of computational power, we're happy to do this, basically for free.
Fantastic.
So anything we can do to help the community and push this forward. So,
Andrew, thank you so much. And just to educate everybody, if you could just tell us a little bit more about the structure and the spike protein, what that is, what it does, and then tell us a little bit about the binding to human cells, if you don't mind.
Yeah, so the spike protein, so when you hear about the coronavirus, you guys see all those things that are sticking out outside the coronavirus. That's actually the spike protein. There is apparently, I don't remember the number, I think it's like 28 epitopes. Those are places that the spike protein can actually bind to the target. And when you compare it to the original SARS to the new SARS, there seems to be a significant number of additional epitopes. Those are the spots on the spike protein that will actually bind. It binds to a region in the body, something that's highly distributed throughout the body. It's called ACE2, which I'm not going to, I know the first angiotensin, but it's ACE2. So that's actually where it binds in the human body. ACE2 is highly distributed throughout the human body. This is probably why it thinks a lot about infection. Now, I should give some background. I'm a computational scientist. I'm a data scientist. I'm not a biologist by training. So I may have said something right now that I could get trolled on. But so forgive me if I said anything wrong. But
we're moving fast. We're going to break a few things, but you've just educated 200 people about spike proteins and the human ACE receptor. So please don't worry. We appreciate it.
Yeah.
Andrew, we're going to move on. And Mike, if your internet is back again, do you want to just quickly finish what you're working on?
Yes. Can you hear me?
We can. Yeah, go for it.
Okay. Yeah. So I'm developing, designing in silico antibodies to bind to the spike protein. So I guess my question is whether we should come to you after we have some initial hits, or can you help us, or can someone help us to expand the scope of our screening to see if our computational predictions lead to effective binding to the spike protein? So I'm looking for like some sort of binding assay like an ELISA, but large scale, to test as many predictions as possible.
Yeah, I would say, certainly one of the biggest areas of interest has been antibodies, and our antibodies team has been working through all the inbound requests we've gotten already. It feels like probably the best way that Ginkgo can help here is by helping those teams pool resources around common screening resources at Ginkgo. So our mammalian engineering team is working on developing the appropriate strategy for expressing those proteins, and then likely running them through the Berkeley Lights Beacon instrument as a way to provide high throughput feedback on antibodies. So given the number of groups that are asking for similar services, we're likely going to try to pool our efforts there and support in that way. I think to answer your question, I think we can help simply via synthesis, if that's quickly supportive to you, and then the screening work obviously takes more in-depth discussions that we're happy to follow up on, but we think there's a lot we can add there. Ginkgo is a platform company, right? So we don't develop any end products ourselves. We work with companies that are developing those products. So I think the other area to partner here is we have a lot of interest on the front end in terms of testing new antibody design strategies. And also we'd like to connect that to a route to market for some of these antibodies as well. So I think there are a lot of opportunities to partner on that side too.
Great. Excellent. Thank you. Thank you, Patrick. And thank you, Mike, for calling in. If you want to ask your question, or you have something to share, or talk about resources that you're looking for, then just raise your hand and I will promote you to a panelist. I do want to give you a sneak peek on something that SynBioBeta has been working on with Leaps by Bayer. Leaps by Bayer is the venture arm of Bayer, and they've been working with us on a very cool infographic looking at the timeline to therapeutic development. So there's a lot of uncertainty about how long this is going to go on, a lot of uncertainty about the testing. If you look at the amount of therapeutics that are on the market, either on the market now as existing drugs that could be reused, or as antibodies, which are going to be the next line of defense, or in vaccines, then you could look at this timeline that we've put together. So this is going to be published shortly, and I'm just going to bring it up. You should be able to see it on the screen coming up shortly. And this is a collaboration, and we've been using the data here from the Milken Institute, who yesterday just published a study showing exactly all the therapies that people are working on. So I'm just going to talk you through this. At the top, we can see there are 7 currently repurposed drugs. That's including chloroquine, the anti-malarial that you've been hearing a lot about in the news. There are 16 antibodies that people are working on, and there's 43 vaccine efforts. So a total of 66 organizations or approaches to treatment and prevention for COVID-19. And this has been a collaboration with Karen Rigel at Leaps by Bayer, and also Jake Glanville at Distributed Bio, and the team here at SynBioBeta. So what you can see here, the repurposing of drugs is already happening. That's happening for compassionate use cases right now, and going through trials as we speak. The antibodies is the next phase, and again, there are some very promising targets out there. I just did a call yesterday that we're going to be recording and putting out with Eric Hobbs, who's the CEO of Berkeley Lights. And I last week did a call with Twist Bioscience, who are both working with Vanderbilt University and Jim Crow there. So we did a video interview with Jim Crow on work that they're doing on antibody therapeutics. They actually took the early patients from Seattle, they flew them down to Nashville, they took their blood, and they've been searching for these B cells that are producing these SARS-CoV-2 antibodies. They just announced yesterday that they have found a number of candidates that are producing antibodies that attack and neutralize the novel coronavirus, and they use the Berkeley Lights beacon in order to do that, to search through tens of thousands of different cells at a time, and Berkeley Lights sent them out a beacon right away. So very exciting news that's coming out from there. And then also Twist, who's been synthesizing constructs for Vanderbilt as part of the DARPA challenge last year on synthesizing rapid vaccines against Ebola. Now it's another target that they've spun up, and within just 60 days, I believe, they've now produced these antibodies. So some very exciting work there. Exciting work that Jake Glanville at Distributed Bio and the team there is doing on looking at the SARS-CoV-1 virus and the antibodies that they'd already created for them, and then using evolution and engineering to evolve and engineer new antibodies to fight the SARS-CoV-2 virus. So very, very exciting work going on in the antibodies world. And what you can see in this chart, we've just done a kind of breakdown for how the three different mechanisms work: the repurposed drugs, the antibodies, and the vaccines. So you'll be able to go to SynBioBeta.com/COVID-19 to get this resource. And if you want to use this or you want to reuse it, then please do. And we'll send this out to everybody on the town hall afterwards. And we'd appreciate when we do your help in publicizing and spreading the word for this. And again, thanks to Leaps by Bayer and everything that they're doing to move this forward right now. Okay, I'm going to stop sharing my screen there, and we're going to go back to the town hall. Ali Savarvi, if you could just introduce yourself and explain what you're working on. Just unmute yourself, and we should be able to hear you. Yeah, go for it.
Can you hear me? Yes, perfect. Hi, everyone. Well, I hope everyone's safe and well as of right now. My name is Ali Safavi. I'm with Gurinova, and we provide like lab automation equipment, but focusing on cleaning devices. And we've been working hard to produce enough devices to offer to the labs during this crisis to deal with the pipette tip shortage. As you guys know, the number of samples is going to continue to increase, ramping up, number of tests going to ramp up, all the studies going on. There's going to be a shortage in the lab supplies, in this case, like plastic consumables, something to like take a preventive measures ahead of time and plan for it. So what we're doing is like, we're working on platforms that can clean, sterilize the pipette tips for reuse purpose for these platforms like TCAN, Hamilton, Beckman, anything like that the labs are using. And we want to help. We've been very successful so far with some of these clinical labs who are doing a COVID testing and reusing the tips mainly like for reagent tips. So we even reduce the risk and be able to implement these like as quickly as possible. So we appreciate everything like you guys do as a community, and we want to be part of like your support team when the time comes like that there's a shortage of pipette tips. You can count on us and our products. So keep on fighting. We got your pipette tips helping you.
Awesome. Thank you, Ali. And if you want to type your email in the chat box, people can reach out to you. I'm just reminded in the chat that, of course, AbCellera has also been developing antibody candidates from patients who had COVID-19. And also, if you have a question, if you just raise your hand, Maya Lockwood from IndieBio, if you want to talk about your announcement, and anybody else, put your hand up and we'll try to get to as many as we can in the next 10 minutes. We have a couple of questions from Gilles Kletter. Are the plasmids with ORFs and antibodies mentioned in the introduction amenable to rapid test development, isothermal PCR, lateral flow strips, etc.? A specialty of my institute, says Gilles. Patrick?
I believe the short answer is yes, but we can provide those sequences to you if you have questions. And again, we're happy to do another round of synthesis to support some of those efforts as well.
Great, excellent, thank you. And Vincent Isabella has a question. For those working on therapeutics or vaccines, can Ginkgo facilitate any collaborations with BSL-3 collaborators to test for vaccine therapeutic efficacy through SARS-CoV-2 challenge? There is a humanized ACE2 mouse model available, I think, in June, possibly July.
Hey, Vinny, how's this going? So short answer is Ginkgo maxes out at biosafety level 2, but we do have collaborators who have biosafety level 3 labs that are being set up for this, including vivariums. Get in touch with me and I can connect you to them. We're working with labs that can support that work.
Great, excellent, thank you. And Stan, do you want to talk about what you're doing? If you could introduce yourself. Stan, are you there? If we can hear you, if you want to speak. So let me see if we've got Tim Gardner. Do you want to mention what you're working on, Tim?
Well, actually, I had first a question, which is
Yeah,
looking
go
at
for
the
it.
timelines on vaccines. I don't know if there's any virologists here or clinical folks.
And Tim is the CEO of Riffin, for those of you who don't know.
Sorry, thank you. The timelines for vaccine development seem incredibly long, and I wondered first why, and secondly, is there anything that the SynBio community could do to accelerate those clinical development timelines for vaccines?
So with the caveat that I'm not a virologist either, it seems like one of the biggest timelines is the important clinical testing work that has to happen. At Ginkgo, we've been thinking about what can we do to accelerate all the other work that needs to go on, assuming that that's stuff that's kind of out of Ginkgo's wheelhouse. So for example, one area where we'd like to start work with companies as soon as we can is process optimization and scale-up development. If you think about, the scale at which these vaccines will have to be produced, how do you even think about deploying these vaccines to billions of doses? That feels like something where this community can get together and help those companies get started on that now. Whereas, personally, from Ginkgo's perspective, I'm not sure we can do much about the clinical trial requirements. That's kind of above, our pay grades, but we can help in terms of process development, optimization, and deployment.
Great, thank you. Tim, you put out a very interesting post on LinkedIn a couple of weeks ago when this got started on looking at risk and how to assess that in your business. Now, I know particularly right now it's pretty uncertain what the future holds. But what advice would you give for startups who are actively, looking around them and trying to decide which brick to step on next?
Yeah. So this is something we put up on March 9th before the shutdowns were happening, because as a company, one of the things that adds to your stress more than anything else is a feeling of helplessness and uncertainty. So the best way to deal with that is to try to take control of the uncertainty. And so what we did is we put together, and I'll post this on the chat here, a risk assessment framework for how do we make decisions under uncertainty. What do we do? Do we stay open? Do we close? And although at this point it may not make a difference in your decisions on whether to close or open or whatnot, what I find is it helps you to understand why you're doing the actions that you're doing and what you should do, and that lowers the stress enormously. It also builds a sense of team because your team understands why you're making the decisions you're making, and they line up behind you. So if you're dealing with stressful situations, I find this type of analysis, step by step, walking through what do you know, what are the questions you have to answer, and putting a framework for making a decision about those questions can be a real help in stressful times. So I just posted the link there, and it walks you through the approach.
Great, thank you very much. Roman Harala has a question in Slovenia. Adgene was shut down last week and writes on their webpage that they operate at reduced capacity. When can we expect to be able to receive plasmids with ORFs in? Patrick, any idea?
Yeah, so they are operating, and we'll likely have an update on their website as to exactly how they're operating. Keep in mind, Ginkgo is under similar circumstances as well. Jason, myself, basically everyone who's no longer in the lab has been working from home for a couple of weeks now. Our lab teams are taking social distancing precautions seriously and spacing out their work and rotating how they come into the lab. So, I think with synthesis starting last week, we can expect constructs to start to be ready in the next week or so. But, please bear with us as we're trying to work around all the restrictions that have taken place around the world, trying to do best practices in terms of social distancing. But that being said, at least for the time being, our synthesis pipeline is operating as normally as it can be, and likewise, Adgene is standing by to distribute those sequences despite their reduced workforce right now.
Great. And a follow-up from Roman says, does Ginkgo have the capability and plans to produce and make available viral proteins for COVID-19 efforts, similar as example for the HIV collection by NIH, available a viral protease for inhibitor screening or spike for neutralizers would be an asset?
I would say yes, our focus is on COVID-19 right now. So all the resources we're making available are specific to that virus, but we're collecting feedback from the community and looking towards further rounds of synthesis. So yeah, please keep those suggestions coming and we'll take that into account as we prepare new libraries.
Great. And we just have a few more minutes left. I want to be respectful of everybody's time. So if we could just do quick 30 seconds, Tom and Pete, and then Howard Salas and Mike Farrow. We'll try to get through as many of these last few comments as we can. Tom, over to you.
Thanks, John. Appreciate it. So Tom Driscoll, I'm with DuPont Nutrition and Biosciences in Palo Alto. So we're interested in leveraging really a world-class protein engineering with large-scale production for particular biologics. It's no secret we've done some work with monoclonals. And for companies like Evolve and Andrew, I'd be interested to see if you have any benchmark data against 3014, 3022 clones, which is a key benchmark of what we're looking at. But if anybody out there has targets that you're interested in process development and scale up, we'd love to talk to you.
Excellent. Thank you, Tom. Appreciate it. Pete.
Hi, this is Pete Carr. You guys getting me? Hi, this is Pete Carr.
Yeah, go for it.
MIT, Lincoln Laboratory, and we are working on applying our cell-free technology, which we call Persia, for doing rapid measurement of different biochemical interactions in particular, and now we're partnering with Ginkgo to scale these efforts up. Essentially expressing cell-free proteins, trying drug repurposing, in other words, the best candidates we can get our hands on for specific biochemical interactions, and trying to get rapid data that way. And while we're starting with small panels of FDA-approved compounds, I would certainly be interested in those who are also doing modeling or development of different protein inhibitors within the family of SARS proteins.
Great. Thank you very much, Pete. Howard Salas, if you want to unmute yourself, and Mike Farrow, if you want to unmute yourself as well, go for it, Howard.
Yes, hi. My name is Howard Salas from Penn State University. And if I could just share my screen for one moment, can everyone see that? Okay. So we developed and are currently validating a new type of massively parallel detection assay for COVID-19 that uses many barcoded RT, reverse transcription primers, so that after you have your patient samples, you can carry out that reverse transcriptase reaction with these barcoded primers. And then after you've synthesized cDNA, you can then pool it all together, carry out a pooled PCR, and then use next-gen sequencing to measure how much cDNA you had in that one pot. And so we see this approach as a way of at least partially overcoming a lot of the diagnostic bottlenecks that are taking place right now. In this approach, you do not need huge amounts of qPCR thermocyclers. You can replace 400 qPCR thermocyclers with 2 or 3 high-seq Illumina sequencers or NextSeq.
Awesome.
So it gives you the opportunity to scale it up. And so we're validating this approach now. We are being very public and we're trying to be as frequent in terms of releasing. We've already released the protocol, already released the primers to carry out the assay, and we'll be releasing the validation data as we go.
Fantastic. Thank you, Howard. And I hope that we'll have some more time later on to go into more depth on this. We'll probably do another town hall specifically on scaling up of rapid testing. So I appreciate you joining the call. Thank you. Mike Farrow.
Yes. Yes, this is Mike Farrow from Tecelligent Biotechnology here in San Francisco. And we're building informatic and computational infrastructure in-house for people who use in-house. So our DNA design team and our machine learning team have been largely pivoted to working with folks who are thinking about building a machine learning model for the fitness landscape for what makes a good vaccine. So we're working with folks at CSIRO in Australia as well as Stanford to think about what would it take to radically reduce vaccine development time by, say, a factor of 10. So Stanford is very good at miniaturizing assays onto microarrays, and we're trying to by the way, the fact that every open reading frame is being expressed as a vector is fantastic. Thank you so much, Jason. This is going to be a wonderful resource. The next step, obviously, is then expressing them in singly and in combination, because it's not just the S1 binding regions, but there are other parts of the vaccine that are implicated, and not just sort of how well they infect the body, but also the kind of immune response that they elicit.
Fantastic.
There's
Thank you,
sort of
Mike.
favorable and unfavorable immune responses.
Thank you for joining us. And just the last two people before we wrap up. Jenny Malloy, you're in Cambridge, UK. What are you working on? How can we help?
Correct. Hi. So yeah, I'm at the University of Cambridge and have been working with the Free Genes Project too. But I also started a not-for-profit called Beneficial Bio. We're supporting startups primarily in Africa at the moment to set up local manufacturing capabilities for reagents and molecular biology enzymes. But we hope that in the longer term that can build some capabilities for local manufacturing of diagnostic enzymes as well. And so we are very keen on kind of any help with synthesis of constructs for expression. We already have the DNA sequences, we just need to put them together at scale. And also anyone who's an expert in protein expression or scaling who really wants to be inventive around how you can do that in a local manufacturing way in a resource constrained lab. I just came back from 2 weeks teaching at the Ethiopian Biotechnology Institute, and we're also supporting a startup in Cameroon at the moment who are almost ready to ship products. So we're kind of keen to have more brains and we kind of see that this could really have a great potential in the longer term, probably not in the kind of current situation in the next 3 months, but in the sort of 6, 12, 18 months timeline and onwards, to really build that local capacity to not have to rely on expensive and scarce imports.
Fantastic. Thank you, Jennifer, for joining us. And finally, Peter Lee at Stratios. What's Stratios working on, Peter?
Yes, yeah, we are an automated cloud lab. So we have laboratory automation in the lines of plate reading. We can do ELISAs, protein kinetic assays, as well as outgrowth experiments. From that, we're able to do a lot of synthetic biology experiments. Sorry, I think there's an echo from my end. Is this okay with you guys?
No worries. Just keep going. We can hear you fine.
Great. Excuse me. So, yeah, we are open for business. We are able to operate with a couple of scientists working on all of our robotic platforms. If we can help with any of the investigations underway.
Fantastic. Well, thank you so much, Peter. And if you want to put your contact details in the chat box, then people can contact you. Jason, final thoughts?
Yeah, just heartened to hear all this today. I think this is an opportunity, and it's good to feel the energy in the community. And congrats everybody on the work they're doing. And let's all stick together on this.
Here. Stay safe, stay strong. Practice your breathing exercise. I really recommend Sam Harris Waking Up app. You can get a free month trial if you drop me an email, and it's very good for reducing your stress. I want to remind you that this too shall pass, and we shall rise triumphant on the other side of this. And it's a tough time, I know. We're all here to support each other. So thank you all for joining. Thanks for everybody who's doing such important work on this and who is contributing. And if you're in need of help, feel free to reach out, and we'll do our best to connect you to anybody in the community who can help you. Thanks again to Ginkgo Bioworks for all that they're doing in this and for this $25 million initiative. You can contact Ginkgo Bioworks, details in the chat below. And we look forward to seeing you tomorrow with the CEO of Moderna, Stefan Bancel, on their new vaccine, and on Friday for environmental sequencing and response to COVID-19. And then next week for Craig Venter and Drew Endy and Jim Collins and George Church. Will all be live doing town halls where you can dial in and ask them their Q&A. And we're also going to be doing a special on Wednesday morning in a virtual environment called Nanome. And we're actually going to be pulling up the COVID-19 structure, the protein, looking at these spike proteins. If you have a VR headset, then please shoot me an email if you want to be invited. It's john.cumbers@synbiobeta.com. There's only 20 people allowed in the nano room, but we'll also be streaming it live on the web so that you can join us on that. If you don't have a VR headset, you can still join in the fun. Okay, we will see you soon. Thanks again, everybody, for joining us today. Bye-bye now.